membrane proteins. They are having a particular adhesive proteins. So as adhesive proteins are going to promote the adherence to the host cell. Then they are producing hyaluronidase, which will degrade the hyaluronic acid present in the tissue lining, and it will degrade the tissue and enters inside the tissue. The third thing is that they have the coating with the host cell fibronectin proteins, and these fibronectin proteins will protect them against phagocytosis processes. And they are also having the tissue disruption primary result from the host immune response of infection. So what happens when they enter inside the tissue lining? By degrading the tissue lining using hyaluronidase, they cover themselves using fibronectin, which they take from host cell, and then the host cell machinery, host cell immune system is trying to go against them, and that in turn further weaken down the disease condition, further amplify the disease condition of the host. Okay. So if we look at the epidemiology that you can find, this Treponema pallidum is having only one host for natural, which is the human. So though there are no reservoir hosts, so the only way of having this disease is the contamination with any human materials. Okay, so the first and foremost thing for that is a safe sex to prevent this kind of diseases like syphilis. Okay, so that's about this Treponema pallidum. Now let us move on to the next type, which is Borrelia, which is also a gram-negative spirochete, and is also having spiral rod-shaped structure. And again, they are possessing flagellas, but instead of 3 flagellas, as we can see in case of Treponema, we are having 7 to 20 periplasmic flagella, and these flagellas are providing them the twisting motility or the screw-like motility from one place to another place. Now they can easily be seen by light microscopy, and as we can see in case of the Treponema, they cannot be visualized through light microscopy, but we can see this Borrelia using light microscopy, but we have to stain them with different types of technique, which is called the GM staining or right staining, instead of the gram staining, as they are still gram negative, but still we cannot stain them using gram stain. We have to stain them using GM or right staining. And they are microaerophilic, that means they also need very fewer amount of oxygen for their growth. So as we can see, most of the spirochetes, they are not oxygen lovers. They some of them are very well when they are low oxygen, like microaerophilic, like Borrelia, and some of them won't mind if oxygen present, but still they generally do not love oxygen and do not want oxygen for their first preference. Okay, and they need complex nutrients. Some species could be cultured but have slow growth rate, as you can see, 18-hour division time. As you can see in case of Treponema, there are 30 hours of division time or replication time. So they are very slow growing bacteria in normal situations, in vitro situations, because we cannot culture them. We don't know what kind of nutrients they need, because they are really very high complex nutrients demanding bacterial species. Okay. Now if we look at the structure of Borrelia using the GM staining, that you can find something like that. So you can see this is the Borrelia, and these are the cells of host which is infected with this Borrelia. Now if we look at this Borrelia using dark field microscopy, you can find something like that. So it's a very so dark field microscopy is being used because of looking at the structural detail or the morphological detail, as you can see here. Now if we make a cross section of this Borrelia, what we can find using the electron microscopy, we can see that this is the inner cell inside the cell, which is having the cytoplasm, and this is the inner cell membrane, and this is the outer cell membrane. In between this inner cell membrane and outer cell membrane, what we are having, the periplasmic space, and one side of this periplasmic space is little bit more illustrated, little bit more stretched than the other sides, and this side is actually creating the periplasmic space. The area for periplasmic space is just getting increased in this particular periplasmic space portion, and this region is full of this flagella. So flagella is just coming out and can be transported from one end to another end throughout this stretched end or illustrated end of uh, this Borrelia, and more and more flagella can pass through this region. So as you can see, all the other sides are free of flagella, but in particular one side, as you can see, which is really stretched out, there are lots of flagella just coming out. So if you look at the flagella, this will come out like this. So above the picture and below the picture. So if we construct the image 3D, okay. And now look at the clinical diseases caused by Borrelia. The Borrelia burgdorferi is a causative agent of Lyme disease. Okay, there are lots of different Borrelia species which cause diseases, but Borrelia burgdorferi causes the Lyme disease. Also the relapsing fever, that means which is simply flu-like symptoms or fever, abdominal cramp, and all these things will come through. And now Borrelia are actually using techniques of releasing endotoxin to degrade the tissue lining, to degrade the cell, to go against the host cell instead of exotoxins. As we know, they are gram negative bacteria, so normally gram negative bacteria can produce the exotoxins, but Borrelia usually do not produce exotoxins. They produce endotoxins for their degradation purposes. Okay, and immune reactivity against the Lyme disease agents may be responsible for the clinical disease. And so again, the actual basis of causing this disease or establish this diseases is lie on the host immune defense. So they produce some materials and they fabricate themselves using the host materials. So host, when they try to go against that, it will just going to destruct the whole balance of the immune system, and that's how the immune system goes down and the disease start to thrive. So that is the way of creating this disease. Now if you look at the epidemiology for this Lyme diseases, then you can find there are louse and ticks which are vectors for carrying this kind of Borrelia burgdorferi bacteria. Okay, in human, when you think about the human diseases, there are body louses which just carry these diseases from one place to another, which causes the relapsing fever. If we think about the Lyme disease, where the reservoir are rodents, deer, domestic pets, and there are ticks of those domestic pets and deers and rodents, we just cause we just carry the bacteria for this Lyme disease, and that's why they need these louse and ticks to complete their life cycle. Okay, and so if we look, this relapsing fever is an epidemic in the Europe last century, and Ethiopia, Rwanda, and in these places this is an epidemic. And we can also have the epidemic of Lyme disease worldwide, considered the learning vector-borne disease in leading vector-borne diseases in US. So this Lyme disease is really one of the most major vector-borne diseases. Now let me go to next slide. Okay. Okay, now we are going to talk about the leptospires. Now, leptospires are also thin coiled spirochetes with a hook at one end or both ends. So this is the differing region. This is the unique feature of leptospires from other types of spirochetes. As we know, in all the spirochetes are very thin. They are having the largest periplasmic space. They are having the intracellular. They are having the periplasmic flagella. But in this leptospiris, what they are varying, they are just varying at the production of the hook in the both end of their body. Okay, and they are also motile by using two periplasmic flagella, and those flagella is just coming out from the inner cell membrane, and it will go from one end to another end. It will contact or attach both ends with each other. Okay, and they are also obligate aerobes. That means they must need oxygen for their growth. So we can see these leptospires are the only one type of bacterial species which are obligate aerobes. That means which need oxygen for their growth purposes. Okay, they grow slowly on medium enriched by the vitamins. They need vitamin B12, B2 and B12 and long chain fatty acids and ammonia for their growth, and they need the temperature of 28 to 30 degrees Celsius for throughout the time for their growth purposes. Now, Leptospira interrogans complex are the human pathogenic strains. This is the type of human pathogenic strains. Interrogans are the human pathogenic strain, as you can see. But still, they need the right environmental situation for their growth. So, though they infect the human body, they need a subsequent amount of time and temperature reliance to finally go and thrive inside the host body like humans. Okay, there are many, almost we can see over 5 lakh severe cases of human leptospirosis around the world, and the mortality rate is 2 to 5%. That's why that makes the leptospirus a very threatening bacteria species. Now, if we look at the structure of this leptospirus, what we can see in the electron micrograph, we can see the structural detail. This is the 3D structure of the electrospirus, and you can see, if we look at the structure using the silver staining, because we cannot see them using normal staining. So if we look at the silver, look onto them using silver staining, and we can find them that they are having the spiral structure, but the end of their body we find the presence of these hooks. Now these hooks are actually helping them to attach with a particular tissue, to invade a particular tissue. They can secrete that hyaluronic acid, hyaluronidase, to degrade the hyaluronic acid and others to degrade the tissue cell lining that helps them to finally enter inside the tissue. Okay, so these are the characteristic feature of leptospirus. Now look at the pathogenesis and clinical diseases of them. Now the virulence they are possessing is dangerous. As we can see, they just cause severe infections. Just for in the first time, the infections just like flu-like or severe systematic diseases. They can produce skin patch, skin rashes. They can also produce the conjunctival suffusion, and with jaundice also. So this kind of dangerous type of symptoms can be possessed by this leptospirus. It will finally make renal failure or meningitis like these symptoms, diseases, and finally extensive vasculitis. The actual thing this leptospirus are intending to do is to disrupt the epithelial lining, or the endothelium lining of blood vessel. So actually what happens when these bacteria start to invade into a particular tissue, for example they start to enter into your kidney, and they just start to invade into the kidney tissue, and what they what they do, due to the damage of the endothelium layer that they cause, what happens, the small blood vessel is just going to disrupt, and what happens, blood is just kind of leak out, and that's how that organs start to deprive of nutrients and all these things. So what happens in turn, if it infects the kidney, you will have a renal failure. If it infects your head, your brain, you will have a meningitis, and this kind of disease is really dangerous. As we know, these are life threatening diseases. When somebody having the renal failure or meningitis, these are the life threatening diseases, and this kind of diseases, that's why are really important. That's why we need to talk about this leptospirosis or this spirochetes, which are devastating. Really, we need to look about them and how to control them. And as we know, this kind of infection can be transferred from other type of vectors into human, and if we are a little bit more concerned about our health, we can prevent this kind of diseases. Now, if we look at the epidemiology for them, then you can find main reservoir are the rodents. So that's why you need to prevent the rodents to stop this kind of infections. So rodents, as we can see, not only in case of Borrelia, but also in case of this leptospirosis, these rodents are the causative vectors for carrying this kind of diseases. Okay, especially rats, which harbor the bacteria in the renal tubules of the rats, and they excrete the bacteria through their urine. So somehow when we are living in the unsanitary conditions where you have a lots of heap of garbages, and rats just coming in and they just urinate on them, and finally if somehow we have any contact with that rat urine, and sometimes when the rat if urinates on the water and we drink that water, and what happens, that can be transferred into us, not only into us but also other in mammals. Suppose your pet can be infected by this, and if you further have contact with your pet, then you can infect through this. So this is an epidemic that can be caused from one place to another place, and finally that come to us. Okay, so man and animals such as livestock or companion animals such as dogs are infected via the bricks and their skin through their mucous membranes. So that's why we need to little bit more concern about rodents and to control this kind of leptospirosis diseases. That's it, and I hope that can help.